The Q stands for a molecule called ubiquinone, or it's also known as coenzyme Q. I'll show you the structure of that in a little bit. Yeah.
Why does complex 2 come last?
So it doesn't really come last. It kind of comes at the same time as complex 1. Okay. It's just a matter. I like being able to show the movement of the electrons from NADH all the way to here. And then we've got this other complex, complex 2, that is also generating QH2. And I said I'd kind of like to be able to draw it out here. You know, I could have gone complex 1, then complex 2, both showing a make QH2. It's just a matter of sort of how I presented it. But it's not really it's not last in the flow of electrons. And I should actually, I'll mention as a side, we'll get into this a little bit. There are other complexes involved in other parts of metabolism that also generate QH2.
Professor,
Yeah.
does that mean that an electron that goes through the electron transport chain doesn't go through 1 and 2, it just one or the other?
That's right,
Okay.
exactly. Yeah, and so the electrons coming here are the ones coming from succinate, basically, and whereas the electrons in this pathway are the ones coming from NADH. Any other questions? Yeah.
In complex 2, where does that FADH2 actually go? Because you mentioned that you only did that because people couldn't see.
Yeah, so maybe another, I'm just going to draw it. I'll draw it really big here. So we have succinate going to fumarate, and that releases 2 electrons. Those electrons are transferred to FAD to make FADH2. And then again, this is enzyme bound. Then those electrons are, as that gets reoxidized to FADH2, those 2 electrons pass through and then ultimately go to Q to make QH2. Is that a little bit easier to see? And what we're going to see as we get into details, this doesn't just pass willy-nilly, but there are actually some specific redox centers in here where the electrons are passed from one to the other. Now you'll see this, you might see this in presented different ways in discussions. So a lot of times texts, particularly maybe the simplified, we'll just talk about citric acid cycle generating FADH2, and then FADH2, contributes into complex 2. But what gets lost in that is, no, complex 2 is that succinate dehydrogenase. All right, let's look at some of the details. Okay, we're going to start with complex 1. So now we're going to go in. That was the overview. We'll get into the specific complexes. One is complex 1. These also have other names. This is also NADH, coenzyme Q, oxidoreductase. So this is pretty well. Let's go, first we're going to look at the structure. We've been talking about all these enzymes, and again, most of the models of the enzymes is that they're usually like either monomers or maybe dimers or maybe even a tetramer, something like that. Pyruvate kinase turns out to be a tetramer. And, but you have these polypeptides, and most of the chemistry is associated with the chemistry of the side chains of the amino acids within the protein. And we kind of blew that model apart a little bit in pyruvate dehydrogenase complex and saying, no, it's actually this complicated thing, and there's lots of these others. So we're going to kind of blow that model even more when we start talking about these complexes. So in this case, there are 46 subunits. This is the eukaryotic system. There's also electron transport chains in E. coli and other things that will have slight variations on this. About 900 kilodaltons. So that's pretty big. 46 subunits. Here's one of the things that's wild about this, and it's amazing all this stuff works, is that, so 46 different peptides have to come together to form a functional complex 1. Remember that in a eukaryotic cell, the mitochondria has its own DNA, but then there's also the nuclear DNA. Some of those 46 are actually encoded in the nuclear DNA, and some of them are encoded in the mitochondrial DNA. So in the mitochondrial DNA, it's going through transcription and translation in the mitochondrial process and making some of those subunits. But then you also have some of those other subunits are being transcribed and translated in the cytoplasm, and then have to be imported into the mitochondria where it all gets assembled together, right? So it's pretty fantastic. The other thing too is there are some genetic diseases that basically are due to lesser function of cytochrome of complex 1. There a lot of, if you have a genetic mutation that results in a lack of function of this, then you're not going to be viable. You're dead, basically, right? But there are some that are still partially viable that you get live births and then a result of various different diseases of different severities, that sort of thing. Okay, so again, it receives 2 electrons from NADH. The next thing we'll talk about is internal electron carriers. So, I showed these arrows flowing through here, right? Well, then it's not showing through these collection of polypeptides, but there's actually different redox centers in here that it gets passed from the other. So what are some of these different internal electron carriers? There is a flavin mononucleotide. And I'm going to show you the structures of these in a little bit. So this is kind of like FAD. Remember, FAD stands for flavin adenine dinucleotide. This one basically only has the flavin part of it, so it's a flavin mononucleotide, and it's bound within that complex. And there are iron-sulfur complexes. And there's a couple of different types depending upon how the iron and the sulfur. So this is not sulfur from cysteines, from the side chain of amino acid cysteine, but actually inorganic sulfur, okay, that combine with iron atoms in a particular arrangement. Now I'll show you those arrangements in a little bit. And then that iron in this like this can be oxidized and reduced between Fe2 and Fe3, and then carrying the electrons around. Okay, so it goes through these internal electrons, and then it donates electrons to coenzyme Q. Let me add just a couple of more points. And then one is it transports four 4 protons from the matrix to the inner mitochondrial space, creating this electrochemical gradient. And the final thing we'll talk about inhibitors. One of the more well-known inhibitors is a compound called rotenone. I'll just tell you the little story about rotenone. Rotenone is a pesticide. It kills fish. So if you're in fisheries management and say maybe there's some junk fish, you got a lake or you got a pond or something, there's some junk fish took over, some non-native, and you want to kill all the fish, you can spread rotenone in there, okay? And there's something about the gill physiology of rotenone that makes fish particularly susceptible to this inhibitor, and so that you can spread it around, and it doesn't necessarily at low enough levels that don't hurt mammals. You can spread it around, and so you can go on YouTube, and there are videos of basically people going in a little boat, throwing out this pellets that has this rotenone in there, and then pretty soon there's all these fish flopping around in there. Lake Davis. Anybody know where Lake Davis is? Heard that story? Lake Davis had a problem with, that's up in the Sierra, had a problem with people throwing in northern pike into it for some reason. Northern pike is this a very aggressive, piscivorous fish. It basically eats other fish. So it would eat the trout and the bass or whatever else was in there. And I don't know why somebody decided, it's not native, somebody decided it'd be cool to have pike in there. So they threw a pike in there. Well, then the management had to get rid of the pike. And so the only way to get rid of the pike is to throw rotenone in the whole lake and kill all the fish in there, and then restock it with the trout and the bass or everything. You hear that story at all, David?
Yeah, that's pretty visceral.
Yeah. Anyway, so rotenone. Let me show you some of the pictures of this complex 1. So here's the structure, and I set this up to show you. So again, down here is like I put on the board. Down here is the matrix, here's the membrane, and up here is the intermembrane space. And so basically what the NADH ends up coming and binding here and transferring its electrons. This green here is the flavin mononucleotide. And then all these other yellow-red things are different iron-sulfur complexes. And then the electrons flow through into here, and in this part of the protein is actually where you get the hydrogen ion transport. And there's a lot of discussion, basically, what the precise mechanism is that results in hydrogen ion transport, and then ultimately it goes to coenzyme Q. Let's let me talk a little bit more of those internal electron carriers. So here again, here's flavin mononucleotide. So this is very similar to FAD, the flavin adenine dinucleotide, because it, except that one would have an adenine linked here. This one doesn't have the adenine linked in here. Again, this is riboflavin, so that you've heard of that as a nutritional supplement in there, riboflavin here. And so again, what it can undo is a series of single electrons, oxidations and reductions. So here, this is the reduced form here. And then it can transfer those electrons down through a series of these different iron-sulfur complexes. And there are 2 main structural types of iron-sulfur complexes: the 2-iron, 2-sulfur. So this is the 2 inorganic iron atoms, or the 2 iron atoms plus the 2 inorganic sulfurs. And then they're also complex with sulfurs that are coming from cysteine residues in here. And then here's just another arrangement where you actually have 4 iron atoms and 4 sulfur atoms in there. And again, what these will have is just these variations in structure give them different redox potentials, so they're able to basically transfer those electrons in the appropriate order down this redox chain through that arm that sticks down into the mitochondrial matrix.
So, Professor?
Yes.
Rather than visualizing the iron or the other atoms moving, it's just the electrons bouncing from one atom to the next?
That's exactly, yes.
Okay.
Yeah, they're like, and so, here, this is somebody where we basically have mapped out those iron sulfur complexes within that arm, and you basically see that, okay, well, it'll just bounce from here to here to here to here to here to here, and kind of go on down.
Okay.
Go on. Okay, so then eventually they go to this ubiquinone, coenzyme Q, Q10. You've seen this. You might have seen this advertised as a nutritional supplement here. And so this is the molecule. Hey, here's a question for you. Going back to that last, what kind of molecule, what kind of lipid is this here? Isoprenoid, right? So you see these isoprene groups here. This is an isoprenoid here, and then it's been modified to have this ring structure here. These two methoxy groups here. This is the oxidized form, and then here's the reduced form. So this is the Q, and that's the QH2. All right. But we'll get back to this later. All right, now we'll go ahead and go to complex 2. This is the other complex that's generating QH2. So this is either known as this succinate coenzyme Q oxidoreductase or succinate dehydrogenase. So structure, this one is a 360 kilodalton, it's a little bit more simpler, homotrimer. So much less complex than, or complicated than complex 1. It receives 2 electrons from succinate. If you look at its internal electron carriers, it has FAD, and that's basically what this is, this FAD right here. And it also has iron sulfur centers. Like complex 1. Then it also has the other one. Here we're going to introduce cytochromes. This one particular one known as cytochrome B560. So this is a heme protein. Now our iron, instead of being associated with a bunch of sulfur atoms, is actually complexed in with a heme.
Professor?
Yes.
What is the letter after KD?
A. Kilodalton, KD or KDA, it's all kilodalton.
Okay.
You know what a Dalton is? Like this. A Dalton is 1 unit of molecular weight. So if something has a molecular weight of 18, water has a molecular weight of 18, you would say it's 18 Daltons. It's like this. It's usually, when you're talking about small organic molecules, you don't use that terminology, but I guess for history or historical reasons, it's used in talking about particularly protein size. And so you'll talk about kilodaltons, because proteins are so big, you tend to talk about kilodaltons. So if a protein has a molecular weight of, a typical protein might have a molecular weight of 50,000. And so the molecular weight of a protein is 50,000, you would say it was 50 kilodaltons. So 360, that's a little bit big. That complex 1, 900 kilodaltons, that's really big. That's a molecular weight of 900 on 1,000. I think the biggest is several million in there, biggest protein. And then you can have small proteins that are just 100 kilodaltons, or 50 to 100 kilodaltons.
Thank you.
Yeah. Okay, internal electron carrier, FAD, iron sulfur complex, cytochrome B5,6 heme. I'll show you the structure of that in a little bit. Let's see what else. Oh, the other thing is that there's no H+ transport. Okay, I'm going to show you the picture of the heme. Before we do that, let's just go ahead and go to there's nothing here I want to get rid of. I'll just Okay, so the next one is complex 3. Which is known as coenzyme Q cytochrome C oxidoreductase. And let's see, again, pretty complicated. And then I'm not asking you to memorize these specific numbers. The main message I want to say is these are structurally complex molecules. This is about a 485 kilodalton homodimer. But each monomer contains 11 subunits. That's really the bottom line. It's like, wow, there's a lot of stuff going on. Okay, receives 2 electrons from QH2, from the oxidation of QH2. Now we generate in either complex 1 or complex 2. It also has internal electron carriers. It has an iron sulfur center. That is known as a Rieske center. And this, what makes this one differ from some of the other ones is it actually has histidines that help coordinate it. I'm not going to get into that detail. And it also has a couple of cytochromes. B562, B566. And then it donates its electrons to cytochrome C, which is a small protein soluble in the intermembrane space. Oh, and then I guess the important thing is it's really what's known as the Q cycle. The Q, we're going to get the details of the Q cycle. It's basically the interaction of QH2 with complex 3, and it's a multi-step process, but basically results in the transports of 4H plus. Okay, so we're talking about cytochrome hemes. And so, again, we have this porphyrin ring structure here, this heme structure, in which you have an iron in the middle. So that makes the cytochrome. There'll be slight variations on the structure on the outside here. And what's that going to do is it's going to change the redox properties of the different molecules to give this iron in here. This iron is actually going to carry the electrons, and it's going to change the redox potential slightly based upon that. And so, let's see, just a sec, do I have that? No. So this cytochrome B, for example, has this structure with its methylene up here. Cytochrome C has this structure here. It's actually connected to the covalently connected to the protein, and these will have slightly different spectral characteristics and with absorption maximum at different wavelengths. So like the B560 versus the B562, that represents specific structures with slightly different substitutions around here that says slightly different spectral characteristics, and they're going to have slightly different redox potentials. And again, so these redox potentials are basically tuned for the energetics that are required for the transport of electrons through the system. Okay, yeah, let me go back. We'll come back to that. All right, complex 1, complex 2, complex 3. Now we're going to finally come to, I'm going to have to get rid of complex 2 here. We're going to get to complex 4. This is known as cytochrome C oxidase. I would say of all these sort of alternate names that I've given you, this is the one that's most commonly used, cytochrome C oxidase. Again, if we look at the structure, it's 410 kilodalton homodimer with 13 subunits in each monomer, or with 13 again, complicated structure. So it receives the electrons from the cytochrome C protein. One electron at a time. Cytochrome C is a single electron carrier. So from NADH, you're going to end up generating two cytochrome C molecules, reduced cytochrome C molecules, that are going to donate its electrons. We look at some of the internal electron carriers. We have some more cytochromes. Cytochromes, they're cytochrome. There's a cytochrome A, a cytochrome A3. And again, these will have slightly different substituents around that porphyrin ring. Then, right at the reaction center, where the actual reduction of the oxygen to make water takes place, there are copper ions. I'm just going to put copper ions. They're known as CUA and CUB. We won't get into that detail. Now the overall reaction is to take an O2 molecule plus 4 electrons plus 4 H plus to make 2 water molecules. Now, you know, when I did this, the overview over there, I was following the path of 2 electrons from NADH. So it's actually going to take 2 NADH molecules to be able to drive this full reaction. This oxygen here, because this is where the electrons from NADH or from succinate end up landing, right? They end up landing on this oxygen to make water. Okay, so this is the terminal electron acceptor. I guess the other final point that we'll make is that we end up transporting 2 H plus, at least that's per NADH. That's how we've been counting it. Let's slide this over here. I guess I can't. Couldn't get totally out of the way. I'll give you another minute or two. So I guess I want to show you one more. Okay, here's the cytochrome hemes. Let's see. Okay, so here's what I want to talk about here. And again, just to show you some of the detail about the actual reaction center here. I'm not going to have you responsible for understanding this level of detail, but I at least wanted to share it with you and expose it with you. So if we look structurally, just because really how fundamentally this important is to heterotrophic metabolism, oxidative metabolism, right? This is what's driving the whole thing, is you have some hemes. This is like the reaction center here. You have some hemes in there. We talk about this heme A and heme A3. We have a couple of copper ions in there that are coordinated with some histidine residues in here. And there's a magnesium ion here, and there's this tyrosyl radical that's actually in here. The whole process basically involves, if we follow this cycle, you bind the oxygen, you bring in several electrons that are coming in from cytochrome C, right? And then ultimately you end up making 2 water molecules through this whole cycle. And the bringing in of those electrons is coupled to the transport of hydrogen ions in this whole process. Okay, so again, I'm not going to have you responsible for the details of this, but I guess kind of wanted to take a little bit of time to pause and reflect that, it's the chemistry of what's going on in that little site. First of all, one point is that what drives this is a lot of inorganic chemistry. I love organic chemistry. We talk about the organic mechanism. We talk about the chymotrypsin mechanism. But here, this one most fundamental process, it's really about these copper ions and these iron atoms that are actually doing the work, right? And that the role of the protein is just basically to get those things in the right place and position the right. It's really inorganic chemistry that is actually driving that process. But you think about, okay, here's the oxygen, right? Coming into this system, going through this reaction mechanism. Think about all the physiology and ecology that drives that process, right? This right here is why we have lungs and a circulatory system, right? Because all of our cells, our brain cells and everything like this, they pretty much need oxygen to be able to survive, right? All of the heterotrophic organisms out there need that oxygen. Why do we have a heart and a lungs and a circulatory system? It's to provide that oxygen to get it to this reaction center. And what we'll see when we start talking about photosynthesis, the ability for this to basically work depends upon the availability of oxygen, right? We have this oxygen availability in the atmosphere. That didn't, if we look at the evolution of the planet, that only happened after photosynthesis got going to put that oxygen in there. And as we'll see when we get into photosynthesis, the photosynthetic electron transport chain generated the oxygen, then all us heterotrophic organisms, now that we have an atmosphere of about 20% oxygen, are taking advantage of that oxygen in this little complex to drive all of that. Why does a fish have gills? Because of this little reaction center right here. Okay, and so what this is doing, oxygen is so electronegative. It wants those electrons so bad that it's basically pulling all those electrons through that electron transport chain, and then coupled to that pulling of those electrons through that electron transport chain is the movement of all these hydrogen ions across the inner mitochondrial membrane, and then we're going to use that gradient, that electrochemical gradient, to drive the synthesis of ATP. Anyway, it's just kind of, fascinating. It's just like, it's this orientation of all these components in this reaction center that just drives so much physiology and ecology and everything. I should also mention that there are, in the absence of oxygen makes a great terminal electron acceptors, but there are alternative terminal electron acceptors, and there are alternative electron transport chains in different microbes. Microbes are pretty fantastic. The one story I like to tell about that is in your I have a boat I keep down in Richmond, and it has a holding tank, and you put the sewage into the holding tank, and then you got to pump it out, right? And if you don't get enough oxygen in that holding tank, then what happens is you get certain microbes that grow that use sulfur as a terminal electron acceptor rather than oxygen as a terminal electron acceptor. And when it generates sulfides, and sulfides smell bad. And so sort of the whole key to keeping your head from stinking and your holding tank from stinking is to keep it oxygenated. Because as long as there's plenty of oxygen in there, then the microbes that use oxygen as a terminal acceptor will dominate, and you won't get any of the sulfides. If you don't have oxygen in there, then these other ones will make your head stink. All right. So this is just another view, the sort of the summary of the whole process here. So through all of this, what we've done is we have taken these high energy electrons, give them to oxygen over here, right? The oxygen loves them. Oxygen's basically sucking the electron through in here, and there's enough energy difference between these electrons here that we can use that to drive the transport of H+. Now the next part of this process is we're going to take this H+ and we're going to let it flow back through this complex, and it's pretty fantastic. Actually, what you end up doing is as those hydrogen ions pass through here, you actually end up making a little motor. It's really a little motor that spins around. As it spins around, it induces conformational change in this part of the ATP ACE. That conformational change is what ends up driving the synthesis of ATP. It's fantastic. It's just really fantastic. So now what I'm going to do, I'm going to take for another 10 or 15 minutes, and what we're to introduce this process of oxidative phosphorylation, the flow of the hydrogen ions back there, and we're going to go back to a little bit of thermodynamics. I know thermodynamics is a tough at the end of a 2-hour lecture, but we're going to go ahead and power through that. A little bit of thermodynamics to see, well, what is really the thing that's driving the force that's driving that F1, F0 ATPase. As you go through this stuff in the homework and all of that, give yourself permission to have a little bit of awe, because it is pretty fantastic. Oh, I should have mentioned a couple other things too on that complex 4. Inhibitor is a complex 4. Things you've heard of, cyanide is an inhibitor of complex 4. That's probably one. What are some of the other inhibitors? Azide, carbon monoxide. Carbon monoxide binds to the heme of hemoglobin. It also binds to these hemes in complex 4. Those are sorts of things. All right. Electron transport. Now we're going to talk about oxidative phosphorylation. We'll talk about the thermodynamics. So we can think about the process of the movement of those hydrogen ions from the IM space to the matrix. Almost like a chemical reaction. That we can basically say we can have our H+ out to the H+ in. We can think of that as a chemical reaction. Now I'm going to make it more general. You can say it could be any compound. We always call it compound A because I'm going to want to get rid of the charge effect at this. A from the outside, A to the inside. And we can say, okay, what about the spontaneity of that reaction? Well, what's our spontaneity, what's our thermodynamic value that we use to address the spontaneity of the reaction? The delta G, right, that gives free energy. So the delta G is going to be RT times the natural log of A in, that's the product, over A out. We don't have any delta G naught prime because it's the same molecule, right? We're not changing the molecules. So this is known as the chemical potential. So let's think about what that really means. All that is a fancy way of saying if we have a lot of stuff on the outside and not very much on the inside, it's going to move forward. Our delta G should be negative. That's all that's saying. It's just like if there's a concentration gradient, it's going to want to go transport across the concentration gradient. But because this is a charged molecule, H+, there's also an electrical component. So the electrical component is Z times Faraday constant times delta psi. What is all of this stuff? Z is the charge of the ion. In this case, with H+, it's going to be +1. The Faraday constant, here's the Faraday constant. And then this is the membrane potential. So there's two things that can affect whether or not hydrogen ions want to move across there. One is just if there's more of them on that side than the other, just the chemical potential. But also it's going to depend upon the charge, the relative charge here. If it has to basically move against an electrical gradient, that's going to counteract anything that is going to be associated with just the difference in concentration. Or if it's going to move with an electrical gradient, that's going to accelerate it. So there's two components. There's the chemical potential and the electrical potential. So the overall potential is known as the electrochemical potential. And it is going to equal, so this is delta G equals RT times the difference in concentration plus this Z Faraday constant delta psi. All right, so let's see what the relative contributions of the electrical potential. So, for example, in a liver mitochondrion, the delta pH, so the difference in hydrogen ion concentrations that's generated by the electron transport chain is going to be about 0.75. You might think, well, that's not very much, 0.75, that's less than 1 pH unit. Remember, 1 pH unit, it's a log scale. 1 pH unit means there's a tenfold difference. If that delta psi was 1.0, that would mean there's 10 times as much. So it's not quite as much as 10 times. Okay, and so what that ends up meaning is our chemical potential. If you go through the math, and figure out this, put a temperature here, now you have your change in, from this, you can figure out the change in this, put some numbers for typical membrane potential that's in here. The chemical potential equals minus 4.4 kilojoules per mole. The electrical potential equals minus 16.2.
Are we
So
still talking about the livers? The liver?
This is still in the liver mitochondria. So this is an example. And what I'm really saying is that this is about, of the overall driving force for this, about 78% is electrical, and about 22% is just due from the difference in concentration. Yes.
So is that for the entire electron transport chain or just the proton motor?
Okay, so the electron transport chain has created this delta pH. It has moved these hydrogen ions across the membrane. And now the question is, okay, so that's going to create a driving force that's going to allow these hydrogen ions to come back across, right? And so like, what component of that difference in pH is more important? The difference just in the concentration of the H plus in terms of, it could be the concentrate, like it could be a glucose, some neutral species, right? It could be just glucose or some other molecule that if you had a difference in concentration, it would want to go like this. Or is it the fact that it's a charged molecule? And what this is basically saying, it's mostly the fact that it's a charged molecule. That what you're doing is you're moving a charge across. And so one of the things I like to say about that is what you basically turn the mitochondrion into is a battery, right? Is where now you have a separation of charge between the inner membrane space and the mitochondrial matrix. And you have a battery. And then what we're going to see when we look at the F1, F0 ATPase is that electrical gradient is basically going to be used to drive a motor, a little electrochemical motor that's going to spin around, and as it spins around, it's going to change the conformation of these subunits that stick down into the mitochondrial matrix, and those conformational changes are going to drive the synthesis of ATP. So it's really pretty fantastic when you think about the overall transformations of energy. Like we start out with, in glucose, we start out with chemical energy, right? In the carbon-carbon bonds, the carbon-hydrogen bonds. And those also get transmitted to NADH. It's basically chemical energy in NADH. But then the electron transport chain is now taking that chemical energy and it's converting it into electrochemical potential that's mostly electrical potential. So it's like now we have an electrical potential. Then this F0, F1 ATPase is going to turn that into mechanical energy of this thing spinning around, and then that mechanical energy is going to use to drive the formation of the phosphoanhydride bond of ATP. So we're back to chemical energy, right? So there's several steps in transformation there, right? The overall efficiency of that process, about 30%, about 30%. So, which actually is not that far different than the efficiency of a car engine, again, turning its the energy in gasoline into kinetic energy moving down the highway, about 30%. Where does all that other energy go? Heat, right? Heat, exactly, in there. All right, we'll go ahead and stop there, and then what we'll do tomorrow, we'll get in and dive right into, okay, how does this F0, F1 ATPase actually work make it spin and then make those ATPs. And again, it's another pretty fantastic story. Hey, just one more heads up before you take out. This exam coming up on Thursday, there's a lot of stuff, right? Don't wait till tomorrow afternoon to get ready for it. And get on top, spend some time this afternoon, this evening, whenever you have time. And also, what was the other thing I was going to say? That pathway map. Work on that pathway map. If you have questions, you can come ask me.
What are the instructions for the pathway map? Is it